Hello, everyone. We're just waiting for a few more people to join. It's just after 1 p.m. in the UK. I'm just going to provide a quick introduction. So first of all, thank you to everyone for joining. I assume more people will be joining over the next few minutes and throughout the afternoon, but we better make a start, I think. So yes, today we're going to hold the first workshop for the Psy-ShareD MRI data resource. This is a project we've been developing over the last 18 months or so in the UK and with international partners. We're almost ready to go operational, so that we think this is a good time to host the first workshop. There will be others next year as well that we are planning now. So can we just go forward? Thank you. Oh, just use that. So just before I do general introductions to the Psy-ShareD team and thank our partners, just a little bit of housekeeping. So this is the outline for this afternoon session, afternoon in the UK anyway. So now we're just going to have an overview really of the project, and that's going to be provided by Professor Veena Kamara and Dr. Simon Evans. We're also going to be joined in the first hour by Dr. Lucia Franco, who is an academic and also has lived experience of psychotic illness. So I think it's very important that we have this perspective in the resource, in the project, and we're lucky to have Lucia joining us towards the end of the first session. At 2 p.m., I'm going to go through a little bit about how the resource is organized, including just some pointers on how to navigate the data catalogs, etc., and also how you apply to access the data. During that session, I'll also be joined by two other members of the team, Sarah Bucci and Ruba Isman, who are going to talk about various aspects of the data catalogs and how we go about data transfer, etc. I think there'll be a break, so I might. Is it 3 or 3:30 that the interactive session starts?
3:30.
3:30. So there's a little mistake here. My apologies. It says 1500 hours, 3 o'clock. 3 p.m. It will actually be 3:30 p.m. So we give everyone a chance to have a little break, get a coffee or some food or whatever they want. And we will have an interactive session that covers data harmonization, validation, and the support tools. So that's the outline for the session this afternoon. But just before I hand over to Professor Kamari, I just want to acknowledge the team. In addition to myself, Dr. Matthew Kempton and Professor Veena Kamari, who I've just mentioned, we have Dr. Jack Rogers, Professor Rachel Upthegrove, Dr. Simon Evans, Dr. Mariana Zuita, Ruba Isman, and Sarah Bucci have all worked very hard over the last 18 months putting this project together, and you'll be hearing something from most of them this afternoon at some point or another. And importantly as well, just at the outset, I'd like to acknowledge all of our partners who come from a diverse range of institutions, both within the UK, Europe, and globally, and also DataMind, who we will be hopefully pairing with going forward in terms of our data access protocols. And of course, the Medical Research Council, who have funded this research and will continue funding it into next year. So that's just my introduction. Did I say about questions? I can't remember. I covered the questions. If you have questions during any of today's or this afternoon sessions, can you please just put them into the chat, and then we will try and answer those in turn at the end of each session. Other than that, I don't think I have anything else to say in terms of an introduction. I hope that was all clear. I'll be back at around 2 p.m. to do the second session this afternoon. But for now, I'm going to hand you over to Professor Kamari and Dr. Evans for the first of our sessions. Thank you very much.
There we go. Hi, everyone. Thank you very much for coming to our first workshop. Before we go into details of what we are doing in terms of our initiative, how to access data, what it's all about, I'll just briefly remind us what we currently know very well about schizophrenia and what we still need to know to just set the scene in terms of why we are doing what we are doing. I'm sure most of you already know schizophrenia affects roughly 1% of the general population, so it's a fairly common condition. It's not a rare condition. And for those who are affected by it, for the majority, it turns usually into a chronic illness. So it has got significant direct costs as well as indirect costs for the people who are affected by the illness and generally for the whole society. It has been considered one of the top 10 leading causes of disability. So it's something that is important to understand better than how we currently do. In terms of risk factor for the illness, well, there are many genetics and environmental risk factors. Coming to genetics, multiple genes are known to be involved, mostly with small effect sizes. Findings so far have been generated mainly from Western samples, and some of the findings may or may not replicate in non-Western samples. Coming to environmental factors, again, there are many risk factors. Generally, effect sizes for environmental risk factors tend to be larger than what we know for individual genes in most cases. Just listing some here: maternal infection during pregnancy, that raises the risk of schizophrenia. Birth complications tend to raise the risk of schizophrenia in the offspring or in the child. More so when there are no genetic factors or no known genetic risk factors. Childhood trauma and abuse raises the risk of schizophrenia. Substance abuse, for example, use of cannabis has been known to raise the risk. Stress, a major risk factor. Migration, psychosocial adversity, further contributions from these. Being born and raised in cities has also been known to raise the risk of schizophrenia for quite some time. And there may be multiple routes that leads to or that raise the risk either alone or in combination. I'll just put them all together. In terms of core psychopathology, the illness, again, I'm sure most of you already know, is characterized by a heterogeneous group of symptoms, mainly positive symptoms, such as hallucinations and delusions, negative symptoms such as apathy, loss of interest, inability to feel pleasure, and so on, and cognitive dysfunction. In terms of our focus in the 1970s, the focus was more on understanding positive symptoms. In the 1980s, focus shifted more towards negative symptoms. And in the 90s, focus shifted, perhaps we focused more on cognitive dysfunction, perhaps led by the findings that it's actually cognitive dysfunction and to some extent negative symptoms that predict functional outcome more strongly than positive symptoms do. In terms of, for those of you who are not so familiar with schizophrenia and starting out, positive symptoms are generally the experiences which most healthy people don't have, but patients with schizophrenia have, such as seeing or things or believing in things that may or may not exist. Negative symptoms are the experiences that most healthy people have, but people with schizophrenia may not, such as feeling pleasure. In terms of treatments, we know that antipsychotic drugs are quite effective in reducing positive symptoms acutely, but they might be less effective in treating negative symptoms and cognitive function. Having said that, about 40% of patients do not respond sufficiently well to antipsychotics. They continue to suffer from distressing symptoms. And for this group of patients, so patients with antipsychotic resistant symptoms, cognitive behavioral therapy has been tried. And shown to produce improvement in about 50% of patients who receive CBT, cognitive behavior therapy. To sum it up, yes, we have treatments in terms of antipsychotic treatments and psychological therapies, but they don't work for all symptoms and they don't work for all patients. We need to continue to find more effective therapeutic approaches than are currently available. And perhaps understanding neurobiology or brain aberrations more clearly would be helpful. Moving from that symptoms and treatments to what we know about brain aberrations in schizophrenia. Well, starting from the seminal study led by Eve Johnston, Tim Crow, Chris Frith, and team showing ventricular enlargement. That was in 1976, and they had used computerized exomal tomography. And they also showed that ventricular enlargement correlated with cognitive dysfunction. There have been many studies in the 80s, late 80s, 90s, and by 2000, we already had about 150 studies or more than 150 studies that had used region of interest approach to look at one or the other brain regions. And looking at the findings, lateral ventricular enlargement, for example, was shown in 80% of the studies that had investigated the structure. So replicability, 80% means 80% of the studies that examined this structure found it to be found an aberration there. And if we go down the list, amygdala hippocampal complex, 74% of the studies showed it. Third ventricles enlargement, 73%. And if you keep going down, by the time you go to cerebellum, it's only about 31% of the studies that examine this structure specifically find it. So the point I'm trying to make to you here is that there is a lot of variation in terms of some studies finding an aberration and other studies not finding. And also, most of these studies are based on data that were generated in Western samples. We need to see whether similar data emerge or similar findings emerge in non-Western samples. And the reason I'm making this point is that outcome of schizophrenia is believed to be slightly different in between developed and developing countries. So there may be biological or non-biological factors that feed into or determine functional outcome, and we need to try and do better to understand those. Fast forward to 2005. By 2005, we have now studies looking at brain aberrations using voxel-based morphometry, so looking at brain abnormality across the brain. And here again, you can see if you look at the x-axis, that shows the percentage of studies that find aberrations in any particular region. And y-axis, you can see the areas that are found. And again, you can see, similar to what region of interest studies had shown earlier, voxel-based studies also show aberrations. And depending on the area, studies may or may not find. The point here again is that there is massive heterogeneity in findings. So now 2024, there have been many studies, but we are still to fully establish the extent to which MRI-based aberrations, whether it's enlargement or reduction, depending on the area, is inherent or related to the pathogenesis of psychosis. What is explained by genetic susceptibility, because we also see certain aberrations, for example, in unaffected relatives of patients with schizophrenia. To what extent is related to cannabis use, psychotic uses, trauma, and other factors. There are studies that are addressing these specific issues, but perhaps what we need is to look at them in the context comprehensively in large studies, so we can see how they relate to each other. We still need to fully establish the location, extent, and progression of morphometric changes during the prodromal period and after the first episode. We need to understand how specific brain alterations in schizophrenia relate to specific cognitive dysfunction is important, because we know that cognitive function is a good predictor of functional and social outcomes. We need to understand whether brain alterations, or at least some of brain alterations, are reversible when patients respond well to drug and to psychological therapies, not just drug, but also psychological therapies. And we need to try and understand whether brain properties predict illness or treatment outcomes. There has been progress, and progress is continually being made. Here, for example, I show you some data from a recent study where they show that in first episode patients, there are changes in the brain over a period of 3 years. You can see that there is caudate reduction, putamen reduction, thalamus reduction, and there is lateral ventricle enlargement. And interestingly, they also show that lateral ventricle volume is associated with worsening negative outcome, negative symptoms, and poorer global functioning. So there are clinical and cognitive outcomes that are related to changing properties of the brain. Another area that I think needs we think needs attention is the concept of accelerated aging in schizophrenia. For a long time, 1887, Kraepelin described the illness as dementia praecox, mentioning early dementia, so a condition where dementia might occur early. And there are data showing that perhaps accelerated aging is true for at least a subgroup of patients. What we need to understand more clearly, is it stepwise aging? Does it happen mainly nearer the time of the onset of illness, or does it continue over a period of time? Is it accelerated aging? Is it exaggerated aging? Is it faster maturation? Also, the possibility that in some cases there might be improvement over a period of time. So there are still questions that we need to answer. There have been research attempts which are data-driven, suggesting that perhaps there are subgroups or subtypes within the illness. And here I show you data. If you look at the top row of images, these are, so the researchers here have applied data-driven approaches to look at brain properties in patients within the first 2 years after their illness. They identify 3 groups, and you can see on the left is the group that seems to have more changes, both in terms of reductions and that is in blue, and red, that is increase. If you look at the lower 2 rows of images, so the first set of images, the data come from a Chinese sample. The second row of images, data come from an US sample. And here is not first episode, it's mid-course. But again, they identify 3 different groups. And in fact, these groups also show cognitive performance that is very different, with the group on the most left also being most impaired cognitively. So it may be that illness, there are subtypes that we need to understand. My final slide, very recently, Jiang et al. Proposed a theory suggesting there are 2 neurophysiological subtypes. And this is based on their analysis of, again, relatively large set of data, again, mainly here, Chinese patients. And they are early in the course of the illness. They find that there are 2 subtypes. For one subtype, illness starts or there is initial gray matter loss in Broca's area. For subtype 2, it's in the hippocampus. And when they follow up, they find that antipsychotics are particularly effective for subtype 1, but not subtype 2, suggesting perhaps there are neurophysiological subtypes. We can use MRI data to identify those. The question, of course, is why some patients fall under subtype 1 or subtype 2, or there are different genetic or risk factors involved. For example, trauma. Hippocampus is very sensitive to trauma, and we know in areas such as depression that for such patients, antidepressants might be less effective, psychological therapies might be more effective. So perhaps trying to identify subtypes would also help to guide and inform treatments where antipsychotics might be more effective, where psychological therapies might be more effective, and where perhaps there is scope to combine the 2 to improve outcomes. So we have learned quite a lot in the last 20, 30 years, but there is still a lot that we need to learn. And hopefully, Psy-ShareD initiative will help researchers all across the world to answer some of these questions, fill these gaps. And now I hand over to Dr. Simon Evans to tell us about the Psy-ShareD initiative. Thank you.
Good afternoon, everybody. So thank you, Veena. Thank you for setting out some of those really important things that we need to consider in order to make progress going forward, to determine the markers and characteristics of psychosis populations. So I'm going to be building a bit on that and introducing the resource, so putting it in the wider context, but talking about Psy-ShareD in relation to what we're trying to achieve and comparing it to previous similar data sharing initiatives and setting out its benefits, basically, and what we hope you'll be able to leverage this repository that we're putting together to really make progress against some of those key issues which Veena just highlighted. So just to mention that as we all well know, conducting MRI studies in general is a challenging endeavor. It's particularly challenging in psychosis populations, of course. Having access to a brain scanner is the first hurdle. There's things like ethics that you need to go through. And of course, as is especially challenging in the current climate, one needs to be able to access the funding and support in order to conduct neuroimaging work. MRI scans are not cheap unless you're lucky enough to have free access to an MRI scanner at your institution. It's very challenging in order to simply even get the funding to conduct this sort of work. And of course, that's compounded by the particular difficulties in recruiting patients, because as anyone who's tried to conduct a psychosis study surely knows, there's a high level of dropout. You need to recruit a lot of participants in order to ensure an adequate sample size, because so many find themselves not able to go into the scanner or tolerate the scanner. There's also the issue of patient heterogeneity as well. So of course, you're necessarily limited to recruiting within the local area and through your local clinical services. So you need to have an effective liaison with clinicians who have the time to support you in your recruitment is a challenge, and you're necessarily limited by the local population that you have access to. What this means is that if you are fortunate enough to have the opportunity to conduct an MRI scanner in a psychosis population, likely as not, you'll end up with an underpowered data set that is not representative of all the different aspects of psychosis, all the different subtypes which Veena just mentioned, because you're going to be able to necessarily restricted in your sample size and access to patients. Nevertheless, a lot of single site studies have been conducted, of course, and a lot of papers resulting over the last 25, 30 years. And a lot of academics' time, clinician time has gone into this. A lot of funding as well, of course, from funding bodies all around the world. So there is a lot of data sets out there but the outputs generated are by and large not especially helpful in that the power, and we recognize this more and more these days, that the power provided by these single site studies just isn't really sufficient in order to get at the key questions that we need to address. But nevertheless, there is this large volume of legacy data out there. So a lot of time and financial investment has gone into collecting, and their potential at the moment is not being fully explored. Often people will conduct a study, run the analysis that they want, and then maybe the funding ends or they move to another institution, and that's the end of it. The data just languishes in a hard drive somewhere. And despite all of these single site studies that have been conducted, we're still not really very clear about what are the brain signatures, what are the morphological abnormalities which really drive psychosis. We don't also have much clarity around how those link to specific symptom profiles. And that, of course, is really important as we come to recognize more and more the existence of different subtypes within psychosis. We don't know much about how they link to, for example, really important aspects such as cognitive impairment, which has a real impact on people's functional capacity. And this is because if you delve into the neuroimaging literature, you're going to see a lot of inconsistent and conflicting findings. The issue of small sample sizes is undoubtedly one of the reasons why this is the case. Patient heterogeneity is a problem. Often the samples recruited just simply aren't big enough to allow researchers to properly account for different, for example, subtypes within psychosis. There's also, of course, methodological differences too, different regions of interest that have been considered, different analysis techniques that have been used. These have evolved over the years. We now have automated parcellation techniques that are really powerful and useful. So the field has developed methodologically over the years. So a lot of those early studies are not so robust in the way that they were analyzed, because they haven't leveraged the more advanced modern techniques that we have available to us now. And of course, it's really important for us to understand what are these neuroanatomical signatures, what are the mechanisms that underlie psychosis risk and link to symptoms. And we need to really understand how these evolve across disease stages, from at risk through the first episode, through to chronic, and then again, as Vida mentioned, what happens across the lifespan as well as people age, medication effects, things like that. We need to have a much better understanding of that and how they link to the specific subtypes and symptom profiles if we are to advance detection strategies and treatment approaches. But of course, one way to try and get around the problem of low power within single site studies is the traditional meta-analysis, which of course many have been conducted. But these are necessarily limited as well, because you are restricted to whatever analyses were performed by the individual papers that you managed to find, and you're just pooling effect sizes across studies. So you can't really account for methodological differences. You can't account for patient characteristics either. So necessarily, any meta-analysis is going to be very limited in how precise it can be in terms of the hypotheses it can test. Also, what's crucial is your ability to conduct specific region of interest analyses is, of course, limited too, because of the methodological differences across sites, because you are limited by whatever summary statistics are available from across all of your contributing papers. And that's really important in regions like the hippocampus and the prefrontal cortex, because we have all of these functionally distinct subregions which might contribute in very different ways to the evolution of the disease and the link to symptomology, particularly, of course, for cognitive impairment. As I mentioned, in prefrontal cortex, we still don't really understand how these very well, we've got a very good idea, we know robustly that there are abnormalities in prefrontal cortex, but what's really the impact of that on the person's cognition? We don't really understand that. So at the moment, it's currently not clear at all what are the specific, regionally specific brain structural changes that occur as the disease progresses from the at-risk state through the first episode and beyond. So more recently then, again, recognizing the limitations of meta-analyses, there have been various multi-site collaborative initiatives that have gained a lot of attention. And Enigma is probably the most well-known of these, but there's also initiatives like Psy-ShareD and various other sort of smaller scale collaborations that tend to involve only a handful of sites. But these have emerged to try and address the limitations of a meta-analysis. So these are collaborating sites coming together to share their data and perform an agreed and so unified analysis on the data that they hold. And most of the time, this is legacy data. So sites offer to reanalyze data that they've collected in the past. Some of these are sort of long-term ongoing studies with very considerable numbers. And when all these sites come together, you can end up with a very impressive total N, even more than some of those already quite large individual center studies. And these initiatives, what they usually do is specify a standardized analysis pipeline. Often they can't specify an acquisition pipeline, of course, because different sites have different scanners, but they often try and employ the same sort of pre-processing and processing steps in order to try and make the results as well harmonized as possible and maximize the power of the inferences that they draw. Often these initiatives involve centers from around the world, so that helps to address the issue of populations and results just being drawn from single populations, and it helps to make it more representative because, as Professor Kamari highlighted, there is evidence of divergence and differences between geographic regions in terms of psychosis risk and links to the brain mechanisms. And in the last few years, or so, just the last 5-6 years or so, there's been some really high profile and really useful outputs from some of these initiatives that have helped to clarify some of the inconsistencies in the literature. The initiatives vary in terms of their size and their global span, but perhaps the most impressive is the Enigma Schizophrenia Working Group, which includes a wide network of collaborators covering all different continents. And for example, this paper from a few years ago, I think it's the largest output from them so far, which is nearly 4,500 individuals with schizophrenia, and there was 39 contributing sites. And it provided some really useful insights. In this case, it's around cortical thickness differences in schizophrenia and how these link to things like medication and symptom severity. So there's no doubt that these initiatives have made big strides in progressing our knowledge, but there's still, we think, much further to go. And one really notable shortcoming of all of these initiatives is that they are not open access repositories. Data is not stored centrally, so the idea is that these sites, there's no transfer of their raw data. The analyses are conducted by each site, and then statistics are provided for a central analysis procedure. And that means that only sites that are able to contribute data have any say or can derive any benefit from these initiatives. So unless you're lucky enough to hold data that these initiatives can make use of, you can't benefit from these resources. So the problem of analyses only able to be conducted by privileged researchers who have access to facilities hasn't gone away. And although there's been a few exceptions recently, most of the outputs from these initiatives are still meta-analyses in their nature. So each site analyzes the data, often using a unified pipeline, which is useful, of course, but then it's summary stats that are provided and so analyzed to provide as the basis of the paper. And this is in contrast to what are called mega-analyses, which is where raw data is provided and then a unitary analysis pipeline is applied to all of that raw data in one go, and then analyzed using that procedure. And this is problematic because, of course, it makes it much harder to account for these diverse symptom profiles, these different subtypes within psychosis that we've heard about. And at the moment, it's actually really difficult for aspiring researchers who are not at major research centers to conduct well-powered analyses and generate papers and test new hypotheses. There isn't really a decent open access neuroimaging resource. There is, some of you might be familiar with SkitsConnect, but if any of you actually try to use SkitsConnect, you'll find that it's really problematic. Most of the time, at least one of the data sources are down. It's not very large anyway. There's only a handful of data sources in there. It's limited in terms of global reach, and there's very little support available. And given the huge amounts of legacy data that's out there from all different sites and researchers all around the world, going back 10, 15 years or so, this is, we think, a real wasted opportunity. And having a properly well-resourced and truly open access MRI depository covering a diverse range of populations and also disease subtypes, and going from at-risk to schizophrenia and so on, would provide huge benefit. It would allow us to actually leverage all the time and investment that's gone into the legacy data that's sitting around, allow us to conduct reliable hypothesis testing, generate new knowledge, and also really support the field as well. It would allow better capacity building, analysis and publication opportunities for all researchers in the field, regardless of their career stage, where they're based, or what resources they have access to. So that's why we've set out to develop Psy-ShareD. And what we are aiming to provide is a well-catalogued database of pre-existing MRI T1 weighted data linked with clinical and cognitive data. We've made active efforts to maximize the global reach. We've got sites spanning Europe and North America, of course, because those are already overrepresented in the literature, but we've made active efforts to reach out to sites in South America and Asia in particular, because there are large studies, large data sets available from those continents, and we've made progress and we'll continue to push forward to find new site partners and maximize the global span of the resource. Psy-ShareD is also broad in scope in terms of the populations, the subpopulations that it includes. So it's not just people with schizophrenia, first episode psychosis, but also bipolar, clinical high risk, schizotypy, experience of childhood trauma, and of course healthy control populations too. And we're looking to expand into other subpopulations as well. And of course, the key thing is that it'll be free to access via a straightforward application process, which you'll hear more about later. Also, what's really important is that for most of the data sets, contributing centers provide their raw data rather than summary stats. So anonymized data is transferred to us securely. There's a data transfer agreement has to be signed off. But we hold the raw anonymized imaging data, and this allows us to perform a unified and rigorous harmonization of all that data, followed by a unified processing and analysis pipeline. And this, of course, saves you a lot of effort too, because we will be applying FreeSurfer to all the data, generating the feature level data, so the full range of cortical and subcortical volumes, cortical thicknesses, and so on, data that FreeSurfer outputs. We also provide image level harmonization too, if you want to conduct VBM. And as I mentioned, all the data sets have well-linked demographic and clinical data. And then for quite a few of the data sets, there'll be cognitive data available, things like functional capacity too in some cases. And we're putting together a really comprehensive and searchable set of catalogs so that you can find what data sets meet your requirements, what their characteristics are, and whether your analyses are going to be feasible. Our aim is for Psy-ShareD to be a really crucial and vital resource for the community that's sustainable and a long-term thing that's continually growing as well. So we'd hope that over time, contributing to Psy-ShareD becomes the norm for people conducting MRI studies. As I mentioned, we're actively seeking new site partners all the time to further boost the numbers and also the global reach. And as evidenced by this workshop, we are really keen on maximizing the accessibility of the data. We really want people to use it and maximize its potential, because the more outputs we get them, and the higher visibility the project has, the more useful it's going to be. So thank you for attending this workshop. We hope you find it useful, and we hope you make full use of Psy-ShareD going forward.